Hello everyone, welcome to EPG Pathashala program in Botany subject. Today I will be talking on a module of plant physiology, namely transpiration in plants. Transpiration in plants account for about of that water that is vaporized at global land surface and 1/8 fraction of this vaporized water at the entire globe. The availability of water is a primary factor responsible for controlling terrestrial plant production at entire scale. Since plants do not have membranes that are permeable to carbon dioxide and impermeable to water, transpiration is an inevitable consequence of photosynthesis, particularly due to stomatal opening that are required for carbon capture as well as for transpirational water loss. And therefore, understanding of plant transpiration process at ecosystem level as well as at the individual plant level is essential. In this module, we will study the learning objectives of transpiration in plants. These are: study the overview of transpiration, study the physiological mechanism of transpiration, study the process of transpiration at a physical aspect, study the modes of transpirational water loss, study the factors affecting the rates of transpiration, study the relationship between transpiration and plant water use efficiency, and the last, study the genes that are regulating transpirational water loss and plant water use efficiency. Leaf transpiration is the process in which vaporization occurs by diffusion of water molecules from leaf chloroplast to the atmosphere via stomata. As you can see in the figure, transpiration is a loss of water in the form of vapor. Water is absorbed by the roots from the soil and transported as a liquid to the leaves via xylem. In the leaves, small pores, which are stomatal pores, allow water to escape as a vapor. In the leaves, small pores, which are stomatal pores, allow water to escape as a vapor. So the question arises, why do plants transpire? To answer this question, we need to address various reasons for this physiological process, and these various reasons are evaporative cooling, accessing nutrients from the soil, carbon dioxide entry, and water uptake. Evaporative cooling is the thermal sensitive process. It is also called as transpirational cooling. As water evaporates or converts from a liquid to a gas at the leaf cell and atmosphere interface, energy is released. This process decreases the internal temperature of leaf, leading to the cooling effect to the leaf. As you can see in the figure, evaporative cooling, which is exothermic process, uses energy to break the strong hydrogen bonds between liquid water molecules, and the energy used to do so is taken from the leaf, given to the water molecules that have converted to a highly energetic gas molecules. These gas molecules and their associated energy are released into the atmosphere by cooling the plant. Accessing nutrients from the soil is another important physiological benefit of transpiration. The water that enters the root contains dissolved nutrients vital to the plant growth, and it is thought that transpiration enhances nutrient uptake into the plants. Stomatal pores are also responsible for bulk of carbon dioxide capture that is necessary for carbon assimilation into the biomass. The disproportionate exchange of carbon dioxide and water leads to a paradox. The larger the stomatal opening, the easier it is for carbon dioxide to enter the leaf to drive photosynthesis. However, This large opening will also allow the leaf to lose large quantities of water and face the risk of dehydration or water deficit stress. Unfortunately, much more water leaves the leaf than carbon dioxide enters for the three reasons. Water molecules are smaller than carbon dioxide molecules, so they move to their destination faster. Carbon dioxide is the only about 0.036% of the atmosphere, so the gradient for its entry into the plant is much smaller than the gradient for water moving from hydrated leaf to a dry atmosphere. CO2 has much longer distance to travel to reach its destination in the chloroplast from the atmosphere compared to the water, which only has to move from leaf surface to the atmosphere. And therefore, the relationship between transpiration and carbon dioxide uptake, which is also referred as transpiration ratio, or also called as plant water use efficiency. Now, plant water use efficiency can be improved by enhancing the carbon assimilation relative to transpiration value, or by reducing the transpiration relative to carbon assimilation. Water uptake is another important physiological benefit due to transpiration. Plants require water for biochemical reactions and cell expansion, which is necessary for growth and development. However, up to 97% of water absorbed by a plant transpired to the atmosphere through stomata. Transpirational water flux is therefore critical to the maintenance of plant water status, maintaining leaf temperature, as well as absorb essential mineral nutrients which are taken from the soils. The water uptake in plant is essentially a transpirational pool which is directed in upward directions. Transpiration rates depend on two major physical factors. The first one is the driving force for water movement from the soil to the atmosphere, and the second one is the resistance to the water movement in the plant. Driving force for transpiration is a difference in water potential between soil and plant surrounding atmosphere. This water potential difference creates a gradient forcing water to move towards areas with less water. With a dry air, there is a greater driving force to move through the plant that fastens the transpiration rate. There are three major resistances to the movement of water out of a leaf. First one is cuticular resistance, the second one is stomatal resistance, and the third one boundary layer resistance. The greater any individual resistance is to the water movement, the slower the transpiration rate. A simple equation of how these factors affect transpiration is transpiration equals to a difference between water potential of a leaf and water potential of atmosphere, this difference value divided by resistance. There are various factors which affect rate of transpiration. The first major factor is plant parameters, in which stomata, boundary layer, cuticle, and the second factor, which is environmental condition, is relative humidity, temperature, soil water, and wind. Stomata play a critical role in transpiration. Stomata are primary control mechanisms that plant uses to reduce water loss as well as allow carbon dioxide entry to drive the photosynthesis. During daytime, photosynthesis is active At the same time, water exits from the stomatal pores as it evaporates, cooling the leaf. When the stomata are open, transpiration rate increases. When they are closed, transpiration rate decreases. In most plants, stomatal density, the number of stomata per unit leaf area, and stomatal aperture, which is opening and closing rate, regulate the rate of transpirational water loss. Leaf boundary layer is a specialized area just outside the leaf surface which plays significant role in transpiration. The boundary layer is a thin layer of still air near the leaf surface. The layer has a least impact of wind speed. During transpiration, water vapor leaving the stomata must diffuse through this motionless layer to reach to the atmosphere. The larger the boundary layer, slower is the transpirational water loss. Plant can alter their boundary layers around the leaves through variety of structural features which are present on the outside surface of the leaves. The cuticle is a waxy layer present on all above ground tissues of a plant and serves as a physical barrier to the water movement out of a leaf. Because the cuticle is made up of wax, it is very hydrophobic, or in other words, water repelling. Therefore, water does not move through it very easily. A thicker cuticular layer on the leaf surface, slower is the transpiration rate. Cuticle thickness varies widely among the plant species. In addition, leaves that develop under the direct sunlight will have much thicker cuticle than the leaves that are developed under shade conditions. Relative humidity is the amount of water vapor in the air compared to the amount of water vapor that air could have hold at a given temperature. A hydrated leaf would have a relative humidity near 100%, just as the atmosphere on a rainy day would have. Any reduction in a water in the atmosphere creates a gradient for a water to move from leaf to the atmosphere. The lower the relative humidity, the less the moisture in the atmosphere, the greater the driving force for transpiration. When the relative humidity is high, the atmosphere contains more moisture, reducing the driving force for the transpiration. Temperature greatly influences the magnitude of the driving force for water movement out of a plant. As temperature increases, the water holding capacity of that air increases sharply. The amount of water does not change, just the ability of air to hold the water. Because the warmer air can hold more water, its relative humidity is less than the same air sample at a low temperature, or it is also a dried air. Because the cooler air holds less water, its relative humidity increases, or it is also called as moisture air. Therefore, warmer air will increase the driving force for transpiration and the cooler air will decrease the driving force for transpiration. The source for water for transpiration out of the plant comes from a soil. Plants with adequate soil moisture will normally transpire at a higher rates because the soil provides water to move through the plant. Plant cannot continue to transpire without wilting if the soil is very dry, because water in the xylem that moves out through the leaves is not being replaced by the soil water. This condition causes the leaf to lose the turgor or firmness and the stomata to close. If this loss of turgor continues throughout the plant will wilt. Stomata are triggered to open in the light so that the carbon dioxide is available for light dependent process of photosynthesis. Stomata are closed in the dark in most plants. Very low levels of light at a dawn can cause stomata to open so that they can access carbon dioxide for photosynthesis as soon as the sun hits their leaves. Stomata are most sensitive to blue light and the light predominating at the sunrise. Wind can also alter the rates of transpiration by removing the boundary layer, that is a still layer of a water vapor reaching the surface of leaves. Wind increases the movement of water from the leaf surface when it reduces the boundary layer, because the path for the water to reach the atmosphere is shorter. Now we will discuss the modes of transpirational water loss. Transpiration occurs during the day as well as in the night. However, majority of transpirational water loss occurs during daytime. Stomata and the cuticle play important role in regulating the rate of transpiration. Let's first discuss stomatal transpiration. Stomatal transpiration during daytime begins with the distinct stomatal opening that results in an increase in the stomatal conductance, leading to a water loss. Stomatal transpiration increases steadily with increase in the stomatal conductance, assuming two physiological conditions that should follow simultaneously. First, the vapor pressure deficit is constant. Second, the boundary layer resistance is nearly zero. Stomatal density, that is number of stomata per unit leaf area, influences water loss, as the stomatal pore area represents principal diffusion pathway for the water loss. A reduced stomatal number is correlated with the reduced transpiration. Stomatal aperture, that is the opening and closing of stomata, is controlled by a cascade of signaling factors. The important stages in a cascading signaling are increase in the cytosolic calcium ion concentration surrounding the guard cell, which activates ATPase enzyme. The activated ATPase enzyme changes the membrane polarization. The hyperpolarized membrane assists potassium uptake into the guard cell that results in a lowering of osmotic potential, increase the uptake of water in the guard cells, ending into increased turgor pressure inside the guard cells. The ultimate increase in the guard cell volume leading to a stomatal opening. Cuticular transpiration. Apart from stomatal transpiration, cuticular transpiration is another important tissue that loses water molecules from the outside surface layers of the leaves, presumably due to higher vapor pressure deficit. Cuticular transpiration varies between plants of different growth and from different habits. The variation in the cuticular transpiration depends upon cuticular structure, thickness, and the amount of cuticular wax and lipid composition. Nighttime transpiration also occurs in almost all C3 and C4 species. This type of transpiration results in 5 to 30% of diurnal water loss in all plants. Nighttime transpiration results from incomplete stomatal closure. Transpiration rate is critical to the maintenance of plant water status, maintaining proper leaf temperature, and essential mineral nutrient uptake from the soil. Stomatal pores are also responsible for carbon dioxide uptake that is necessary for carbon assimilation, which results into increased biomass and yield. And therefore, the relationship between transpiration and carbon dioxide uptake is referred as transpiration ratios. This is also called as plant water use efficiency, and which can be improved by increasing the carbon assimilation relative to transpiration. Plant water use efficiency can be also improved by reducing transpiration relative to carbon assimilation. Genes are regulating transpirational water loss as well as plant water use efficiency. There are overall 8 to 20 genes which are present in Arabidopsis and many other plant species that have identified which can regulate water use efficiency and transpiration ratios. For example, in Arabidopsis erecta, which encodes a leucine-rich repeat receptor kinase that regulate water use efficiency. This is the first gene identified by using quantitative trait loci mapping approach that regulate water use efficiency. Another gene from Arabidopsis, that is ABC transporter, mediates stomatal opening and closing through abscisic acid, calcium and auxin dependent pathways. A loss of function in this gene by mutation result in a significant reduction in a stomatal opening in response to leaf, which reduces the transpiration. The other genes, for example, Hardy, DREB1 and GTL1 have been shown to be a key regulator in stomatal functioning, leading to a tight regulation in plant water use efficiency and by improving plant water use efficiency in these plants. Let's summarize the topic. Plant transpiration can be determined by vapor pressures of water from inside to the outside of the leaf, as well as from the resistance values, which are reciprocal values of conductance from the inside to the outside of the leaf. Resistance to the water movement includes stomatal, cuticular and boundary layer. The relationship between transpiration and the amount of carbon assimilated is also called as transpiration ratios. This is also referred as plant water use efficiency. Plant water use efficiency can be calculated as the amount of water transpired divided by the amount of carbon assimilated. The primary route of water and carbon movement inside the plant is primarily by stomatal pathway. Beyond this primary stomatal pathway, the cuticle and boundary layer often provide resistance that may influence plant water use efficiency. Plant water use efficiency can be achieved by increasing carbon assimilation relative to the plant transpiration, or by reducing plant transpiration relative to the carbon assimilation. Natural variation in plant water use efficiency is often negatively correlated with the transpiration, but not with carbon assimilation. Therefore, plants have various physiological mechanisms that may influence plant water use efficiency, which is under genetic control. Therefore, plant water use efficiency can be achieved by genetic introgression of genetic determinants or the genes that regulate the physiological mechanism of plant transpiration. Thank you.